Actually, we're going this way, so it didn't happen. Shades, transcription, and prokaryotes. I have to check. Is it recording the internal sound? Audio setting, microphone, right, fine. Okay, close this. So we are studying transcription in prokaryotes. Alright, this is transcription in prokaryotes. What is the meaning of transcription? When there is synthesis of RNA from what? From DNA. That is your transcription. So like if you want, I can draw this. Suppose this is your DNA. This is something like, I mean, not me perfect, I'm not blah blah blah. So this is your DNA, and what happens, this is double stranded, but after transcription it changes into 5 prime. Suppose this is 5 prime, this is it, all the bases are just here, blah blah blah, and this is your DNA and it changes into RNA. Alright, and it can be of 3 different types. It can be mRNA, it can be rRNA, which is your ribosomal RNA, or it can be tRNA. So it can be messenger RNA, it can be ribosomal RNA, it can be transfer RNA. So we can also discuss about one thing, which is your central dogma of molecular biology. What happens, like DNA, like DNA to DNA, that is your application. When we have DNA to RNA, that's what we call transcription. And when we have RNA to protein, changing one form of information to another, blah blah blah, that is translation. All right, that is the thing. Now let's talk about RNA polymerase, and remember that this is actually transcription in prokaryotes. So let's talk about RNA polymerase. This is your RNA polymerase. What happens in it? Actually, we have different types of RNA polymerase. We have 2 alpha, and one is beta, and another is beta dash, and then sigma. So we have like beta, we have beta dash, we have alpha, another alpha, omega, and sigma. So let's talk about the sigma factor first of all. Sigma factor, it does 2 functions. Number 1 is that it scans the DNA, and then it recognizes promoter. It will scan the DNA, like this is the DNA. It will always scan the DNA and it will recognize the promoter and terminator region. So sigma will recognize the promoter region. That is the thing. Let's talk about beta and beta dash. So beta's function is to polymerization of nucleotide, and beta dash function of beta dash is it has helicase activity. That means unwinding of DNA. Like this is the DNA, and this opening of DNA is done by beta dash, and beta is just for the polymerization. And then we have alpha, 2 alpha. So function of is to direct the DNA to be transcribed. Like this is the DNA. Na? So like which part, like starts from my end still here. So which part of the DNA has to be transcribed? So it detects that part of the DNA which has to be transcribed. Suppose this part of the DNA has to transcribe. So this is this work is done by this alpha. And we also have omega, and but we don't know the function of omega, that's unknown. And one more thing is that what I'm talking about, this when sigma factor is attached, like you have beta dash, alpha omega, but when sigma factor is attached to this particular thing like this, so we call it what? This is called holoenzyme. I have a horrible handwriting. Alright, holoenzyme. So now we will stop this. If it's I don't know, some kind of glitch, horrible glitch. Okay, stop.